Biofilm? What's biofilm? Is it a form of biology?
Let me show you a video.
Hi, my name is Loliwen. Me and my group mates would like to talk about antibiotics and chlorine resistance in bacterial biofilm of hospital water systems. These are my group members, Ko Yi Ting, Lai Wei Xin, Li Mingxuan, Lin Zixing, and Nema Devi. Microbiofilms are also known as biofilms. They are the aggregation of microbial cells, which are associated with the surface in almost an irreversible manner. For example, cannot be removed by gently rising. They are attached with a biotic or abiotic surface integrated into the matrix that they have produced. And a custom biofilm provides favorable conditions for genetic material mobility between the cells and has a defined architecture. It is also reported that these surface-associated microorganisms possess a definite phenotype with reference to growth rate and gene transcription.
Where are biofilms found? On the dental plaque. Dental plaques form on the teeth and create a sticky film that can lead to tooth decay and gum disease. River rocks and streams. Biofilm can develop on the surface of rivers, rock, and other submerged objects in aquatic environments. On medical devices, biofilm can form on medical implants, catheters, and other healthcare devices. Wastewater treatment in plants. Biofilms are used in wastewater treatment processes to break down organic matter and remove pollutants from sewage.
Pipelines. In the oil industry, biofilms can form on the interior surfaces of pipelines, which can lead to corrosion and reduce efficiency in transporting oil. Food processing equipment. Biofilms can form on the surface of food processing equipment such as conveyor belts, pipes, and tanks. In soil, microbial biofilms are present in soil and play essential roles in soil health and nutrient cycling. In cystic fibrosis lung infections. In the lungs of individuals with cystic fibrosis, a thick and sticky mucus can provide an ideal environment for biofilm formation by the bacteria.
Hi, I'm Yiting. I'll be talking about the factors of formation of biofilm. The formation of biofilm is based on two factors, which are the environmental factors and structural factors. One of the environmental factors is nutritional cues. Low nutrient condition will create a stress onto the microorganism and trigger biofilm formation. However, the biofilm are not able to mature if the nutrient level are constantly low. Besides, changes in the oxygen level can affect the formation of biofilm. Under an oxygen-limiting condition, the cellular respiration is impaired, triggering the programmed cell death in bacteria. As a result, the bacterial cell lives and releases proteins and DNA into the surrounding, suggesting that the hypoxia does indeed mediate the formation of biofilm. The temperature fluctuation can also influence the production of biofilm. The temperature has a varying effect on biofilm development in different bacterial species. For example, the optimum temperature for Salmonella was found to be 30 degrees Celsius as there is a rapid planktonic to biofilm transition at this temperature. The change in surrounding pH level can also contribute to the formation of biofilm. Neutral conditions favor biofilm formation at 35 degrees Celsius with only limited formation at 37 degrees Celsius. Under an exceeding growing environment, more biofilm are formed at 37 degrees Celsius as compared to the 35 degrees Celsius. Additionally, exposure to antimicrobials, antibiotic with concentration lower than its minimum inhibitory concentration, which is MIC, can mediate biofilm formation. Furthermore, one of the structural factors is EPS. The synthesis of EPS is a self-produced, energy-demanding process attributed to the select condition in the environment. The density, water content, charge, and mechanical stability of biofilm cell are all affected by EPS. EPS directed changes onto the aphasic properties determined by the biofilm cell condition of life in the microenvironment. Flagella is another structural feature in the production of biofilm because it gives the bacteria their motility and aids in the rapid spread of bacteria over contacted surfaces. Lastly, pili are important hair-like appendages that can impact on the formation of biofilm in gram-positive and gram-negative bacteria. In the case of gram-positive bacteria, the absence of pili can lead to the prominent defects in biofilm formed by Enterococcus faecalis mutans. In the case of gram-negative bacteria, the absence of pili in Salmonella would correlate with an impaired development of biofilm. This suggests that the biofilm cannot be formed in the absence of pili.
Hey, just now I saw the first slide talking about the antibiotic chlorine resistance. What is it?
Just continue to watch.
Let's talk about the factors contributing to antibiotics and chlorine resistance of biofilm. One of the factors is the extracellular polymeric substance, which is EPS. Biofilms produce EPS, which is a complex matrix of polysaccharides, proteins, and DNA. EPS acts as a physical barrier that limits the penetration of antibiotics and disinfectants into the biofilm structure. This protective matrix enhances the resilience of biofilm-associated bacteria. The next factor is microbial heterogeneity. Biofilms often consist of diverse microbial communities with varying phenotypes and metabolic activities. This heterogeneity results in different subpopulations of bacteria exhibiting distinct susceptibilities to antibiotics. Some cells within the biofilm may be in a state of dormancy or reduced metabolic activity and hence rendering them less susceptible to antimicrobial agents compared to actively growing cells. Besides that, quorum sensing is also a factor contributing to antibiotics and chlorine resistance of biofilm. Bacteria within biofilms can communicate through quorum sensing, which is a process involving the production and detection of signaling molecules. Quorum sensing enables coordinated gene expression and the activation of resistance mechanisms in response to environmental stress, including the exposure to antibiotics. This communication facilitates collective biofilm response and enhances the overall resistance. Not only that, slow growth and metabolic inactivity is also another factor contributing to antibiotics and chlorine resistance of biofilm. Biofilm bacteria often exhibit slower growth rates and metabolic inactivity compared to their planktonic counterparts. Antibiotics commonly target actively dividing cells, and the presence of metabolically inactive or slow-growing cells within biofilms, they can reduce the efficacy of antibiotics and hence contributing to the resistance.
So, how does biofilm transmit infection? Bacteria can reach the new host either directly by contact with the infected person or indirectly due to airborne contamination, consumption of contaminated food, or contact with surfaces. Basically, bacterial biofilm are aggregates of bacteria attached to surface and each other and embedded in a self-forming matrix, which is composed of substances such as protein, polysaccharides, and eDNA. In addition to the protection provided by the matrix, bacteria within biofilm can employ multiple survival strategies to invade host defense system. By remaining dormant and hidden from the immune system, that can cause local tissue damage and later acute infections. It then also causes an infection by the spread large number of cells. Also, hospital distribution system can act as a reservoir for waterborne opportunistic pathogens, and patients can be exposed to these bacteria through activities such as souring.
In order to control the growth of biofilm, one of the strategies is by using an anti-biofilm substances, which possess several characteristics including antibacterial activities, able to penetrate through EPS into the cells, having unique structure, able to interrupt bacterial cells communication, and works well with other antibacterial agents to enhance its effect. Another practice for hospitals to reduce the growth of antibiotic-resistant biofilm is by maintaining the chlorine level in water distribution system for disinfection purpose, but not exceeding 5 ppm, which is the maximum level of free chlorine in drinking water, so that the water is safe for consumption. One example of anti-biofilm agents is antimicrobial peptides, which is able to interact with the negatively charged components on bacterial cell membrane due to its positive charge. Specifically, lipoteichoic acids and lipopolysaccharides are involved for gram-positive and gram-negative bacterial respectively. Most of the AMP are able to target on antibiotic-resistant bacteria, which can act on different stages of biofilm development through different mechanisms such as forming pores on membrane, which can kill the cells, inhibiting cellular activities such as synthesis of DNA, acting on the substrate surface to prevent adherence of bacterial cells and remove preformed biofilms, disrupting transmissions of QS signals, and lastly, downregulating the genes for mortality.
So, do you understand more about biofilm now? I guess.